So the genes are sort of whirring away, we're getting proteins made, and then microRNAs kind of come in and they sort of stabilize things and dampen things down and keep things as they should be. They sort of fine-tune the system. And that's the idea behind the title of the book, Fine-Tuning Life. They're sort of nature's fine-tuners.
Fellow Homo sapiens, my name is Torie Robinson, and welcome back to Epilepsy Sparks Insights. The RNA revolution has given us an entirely new, exciting way to treat, or at least look at treating, many diseases, including the epilepsies. In today's episode, and part 2 of 2, David Henshall conveys his passion and excitement for microRNAs and their regulation of the human genome, talking about his new cool book, Fine-Tuning Life, a guide to microRNAs, your genome's master regulators. Please don't forget to share your thoughts on this episode with us in the comments below, because I really enjoy reading your thoughts and responding to them. Do subscribe so that we can educate and empower way more people affected by the epilepsies around the world, and indeed more clinicians too with patients who have an epilepsy, so that we can all provide the best care possible.
My name is David Henshall. I'm a professor at the RCSI University of Medicine and Health Sciences, which is a big medical school in the heart of Dublin in Ireland. And I do research on epilepsy. The area I'm most interested in is understanding what happens to genes in the brain during the development of epilepsy, and that's what we do research on, trying to find new ways to treat epilepsy.
Fine-Tuning Life, a guide to microRNAs, your genome's master regulators. So that's quite an enticing title. What's the purpose of your book, and who is it targeted at?
The purpose of the book is to tell the story of these remarkable types of genes that we've been working on for many years now, called microRNAs. So to try and tell the story of them, I think we think that they're fascinating genes. They're a bit different to the type of genes people typically are doing research on. And the story of how they were discovered, how we learnt what they do, how they work, how they evolved in the first place, and what they do within the cell, I think is an exciting story that I thought I had reached a point that I could tell. And because it links to the work that we've been doing on epilepsy over the last 15 years or so, I could see that you could tell the story and move it towards the areas that I'm, of course, most interested in, which is what these genes do in the brain, and more importantly, what they might be useful for in epilepsy in terms of explaining what can cause epilepsy and maybe targeting those new types of genes as a new way to treat epilepsy. There's even a little bit of a story that they could be useful for diagnosing epilepsy as well. So the book begins with a description of the gene pathway, really sort of takes you from DNA to protein, and then tells you where it is that microRNAs sort of act in that pathway, because they sort of work at a particular point, and they work by sticking on to messenger RNA and reducing the amount of the messenger RNA. So essentially they dampen down gene activity within the cell. And they're an important fine-tuning system, so they sort of stabilize gene activity within the cell. And experiments have shown that without this molecule, gene activity becomes rather unstable and slightly chaotic. So microRNAs work as a sort of negative feedback loop to stabilize gene activity within cells. And they evolved hundreds of millions of years ago alongside our protein coding genes.
Uh-huh.
And so they're sort of there the whole time, looking after things, keeping things stable, keeping watch over the gene pathway.
They're mates.
There's a rather nice analogy that sometimes people use to describe microRNAs, that they're the conductors of the molecular orchestra.
Oh, I really like that thought. I went to a classical concert recently. I love some classical music. And it's so true. Like, unless you're all following the same hymn sheet and following the rules, things so easily fall apart and just sound dreadful. And so it's the same within one's body, right?
Exactly. You've sort of got this molecule that in using the music analogy, can speed things up, slow things down, make things louder, make things quieter. So the genes are sort of whirring away. We're getting proteins made, and then microRNAs kind of come in and they sort of stabilize things and dampen things down and keep things as they should be. They sort of fine-tune the system. And that's the idea behind the title of the book, Fine-Tuning Life. They're sort of nature's fine-tuners.
What has been the impact on us as a species over the past sort of 10, 20 years with a better understanding of RNAs and the impact on people affected by the epilepsy on the brain, on us as a species, really?
There's been an enormous, there's been what people have described as the RNA revolution, which is that RNAs now represent a completely new way of treating disease, not just brain disease, of course. I mean, the COVID vaccines are the best example of this, or the most obvious example. But we can now target RNAs to create very precise types of gene therapy, where we can design molecules that will lock on to specific RNA, stop it from working, or activate it, make it work better. And by doing that, you've really got a single gene sort of precision therapy. A lot of the drugs that, well, really almost all the drugs that we use for epilepsy act very broadly. You know, they dampen down brain excitability. They sort of jam things in the neuron to neuron communication, quieten things down, but they quieten things down everywhere. They don't know to just go to this one place and interfere there.
Cheers, mate. Yeah, thanks. Dampen down my life. Great.
So the promise of RNA therapy is that you can target in very, very specific ways a molecule inside a cell, stop that from working or boost it from working, leaving everything else on its own. So you, in theory, would get less side effects. The downside of these types of RNA therapy is that the drug, if you like, to target that is a large molecule. It's complex. You probably can't just take it as a pill. It might be something that has to be sort of injected somewhere, and perhaps injected even into the brain itself, because otherwise it won't get into where it needs to be.
And by big, do you mean, sorry, are you talking like a centimetre? Are you talking like a couple of millimetres?
We are still atomic scale here.
Right.
We're still at the atomic scale.
So why can't that be like absorbed in another way then?
We have this barrier that separates the blood from the brain, called the blood-brain barrier. So basically the blood cells that make up the vessels in the brain are really tightly glued together, and things just can't pass through them very easily. So small molecules can get through pretty well, but these big molecules that we're designing, and in a way what we're designing is something that looks a little bit like DNA itself. So these are very large molecules, and they won't pass simply from sort of the blood into the brain. So we have to figure out ways to get that therapy into specific brain cells, and people are working on solutions for that.
Well, I'm just thinking personal perspective. If you want to shoot me up with a little bit of a couple of cells into my cerebrospinal fluid versus ongoing seizures, I mean, that's just me.
That's another benefit, we think, from RNA therapy. So the molecules that we're able to design last a really long time. So unlike your typical anti-seizure meds that, you have to take sometimes several times a day, most of these molecules that we're talking about now might last 2 to 3 months after they've been injected. So the route to deliver them might be a bit invasive, as you say, intrathecal injections. But once injected, you might not need to have a treatment for 2 to 3 months. The molecule itself survives a really long time in the body. The body can't break it down because it's a sort of an unnatural molecule, and it's big and it's difficult to get its hands on.
So you say unnatural, that might scare a few people. Could you reassure us, please?
So what I'm saying when I say unnatural is that if we design a gene therapy out of, say, pure RNA, it would be digested immediately. We have enzymes sitting everywhere that break down RNA, because viruses use RNA to get their job done. So we have evolved very good defenses against that. So if you like, naked RNA will be dissolved very, very quickly as soon as it enters the body. So what we have to do is make little chemical modifications to the RNA so that our natural defenses don't detect it or can't break it down. So when I say an unnatural molecule, essentially we're taking a sort of a natural molecule, just making a few chemical changes to it so that it evades the body's defense and can survive for a long time.
And so at the end of, say, 3 months that you spoke of before, so, it could have beneficial impacts for that long, what happens to the actual RNA that you've shot into somebody's system?
So we think over time the sort of the artificial molecule begins to break down, that although it lasts a long time, eventually it is sort of removed from the cell or breaks into smaller pieces over time.
And what else would you like to tell us from your book?
So the first chapter of the book tells the story of the normal gene pathway from DNA to protein. And then the moment about 30 years ago where a new type of gene was discovered. That gene would eventually get the name microRNA. So it tells the story of how this gene was discovered, which is a really cool and exciting story because the researchers that were working on the discovery were studying actually how worms develop. And worms are obviously very simple creatures. This particular worm has only about 1,000 cells in its whole body. We have trillions. So it's very simple.
Wow.
But because it's so simple, you could understand which genes switch on and switch off as the worm developed. And they made this discovery that there was this other type of gene that clearly didn't code for a protein, but was really critical for the worm development. So they made this discovery, and then it got largely ignored because people didn't really grasp what had been discovered.
What a big deal it was, how cool it was.
A few years later, a second type of this gene was discovered, and then the big moment was that, that second type of gene is also in humans. So this isn't just a oddity of a worm. We have the same gene. And it was identical. The sequence was identical. So we have been separated. Our last ancestor that we shared with the worm is sort of 600 million years ago or something. We have the exact same sequence of this particular gene. So we have required this gene over the hundreds of millions of years that we have evolved from the last ancestor with this worm. So that's the opening chapter of the book. And it's a really cool story. It's cool because it shows that sometimes you don't even know what you're working on and how it might impact science in the future.
And how exciting is that? It's like the unknown sometimes of the significance of one's discovery, right? That's one of the amazing things about science. Anyway, sorry, next chapter.
Yeah, so chapter 2 is sort of the explosion of interest in this new type of gene that within a couple of years had been referred to as a microRNA. And the remarkable thing was within a very short period of time, 2 or 3 years, scientists around the world had figured out the species that have these microRNA genes, more or less how many genes that we have. There's about 2,000 of these in the human genome. They had figured out the pathway by which they were made, sort of how they got formed, where they acted, and what they did in the cell. And so there's this wonderful sort of explosion of research that generated the core understanding of the whole microRNA world. And it happened within 2 or 3 years.
That's amazing. And which years are we talking about here?
So the kind of explosion moment was around the year 2000 to 2004. So there's this sort of 3 or 4 year period when a lot of the fundamental discoveries were made. I mean, we've continued to make discoveries about microRNAs ever since, but there was this amazing period of time that a lot of the basics were uncovered.
And what was the next stage, the next step, and the next chapter?
Yeah, so I during the next couple of chapters, I sort of explain why microRNAs evolved. So what is it? What is the important job that they do for the cell that wasn't getting done just by our regular protein coding genes? So this is sort of this idea that they work to stabilize gene activity within the brain and within other parts of the body, that they're critical for how we develop, as cells divide, particular microRNA genes switch on and switch off. And essentially, they're critical for every stage of the development of organisms, but also once we've finished developing, they help stabilize the genes that are switched on a day-to-day basis and keep everything working smoothly.
It sounds like they are at least partly responsible for the complexity of us as a species.
This is exactly right. And one of the points I make in the book is about something called the and this is going to sound a bit nerdy.
Please.
The G-value paradox. Right. Well, the G-value paradox is that we share the same number of protein-coding genes as that worm did. We both have about 20,000 genes. So clearly, complexity is not simply a function of the number of genes that we have. So there must be something else in our genome which is taking these 20,000 genes and creating the rich diversity that we have as humans and also other more complex organisms. And this is one of the key roles that microRNAs do. They can sort of take this basic set of instructions, our protein coding genes, and they can use them and control them in ways that creates enormous diversity. All of the different cell types, systems that we have, tissues, all the way up to the wondrous complexity of the brain, is partly helped by the presence of these microRNA genes.
Do you talk about the not so positive aspects of some microRNAs in the book?
Yes. So because microRNAs are so critical to normal, healthy cell function, it's not surprising that if levels of microRNAs either increase too much or decrease too much, then things can go wrong. In the book, I focus mostly, I think, around epilepsy and trying to describe a little bit of the research where we've identified some microRNAs where levels are either too high or too low within the brain. So if levels of a particular microRNA are too high, you can imagine that may switch off genes that we want on. And if levels of a microRNA are too low, then genes that should be off start to switch on. So for sure, we know that changes in microRNA activity in the brain could be contributing to some of the epilepsies. Now, a lot of that research is still at a preclinical stage. So the proof, if you like, is at a preclinical stage. So people have shown, for example, that if you have a knockout, if you remove one or two of the really important microRNA genes, it can cause an epilepsy.
Okay.
And if you stop them getting produced in the brain, then say you stop them being produced in the brain of a mouse, that mouse actually, over a period of weeks, begins to develop seizures.
Huh.
So we know that the brain must make a number of microRNAs all the time, because if you prevent them being made in the brain, then seizures can develop.
Does that happen sometimes? So somebody's microRNA can change and they start developing seizures?
This we don't know. We can study, for example, animal models of epilepsy, and we can see that there are changes in microRNA levels as epilepsy is developing,
Right.
Some of them switching on, some of them switching off. So we think that that's contributing to the development of epilepsy, but proving that happens in a person isn't possible at the moment. The proof may be, if we can design a drug against one of these microRNAs, and that if that can interfere with an epilepsy in a human, then of course then that's more evidence. But at the moment, the evidence is really just coming from what we can do in preclinical models.
Do you talk about what the future may or may not hold in your book?
Yeah, so the final chapter of the book looks to the future and asks some of the questions, what is next for this field of microRNA research? And I talk a little bit about where I think the possibilities and opportunities lie in the field of epilepsy, but I also take a bit of a step back and say, what are the really big discoveries, and what are some of the top scientists around the world that are working on microRNAs? What are they doing in the future?
And do you touch at all upon, actually, in the book, I don't know if it'll be at the end or in the middle, throughout, but lots of people I kind of get a little bit scared. You talk about things in acronyms that we're not aware of, and talk about anything to do with genetics. It's like, oh my goodness. So do you cover anything about the manipulation of this data or people using microRNAs in potentially a not so positive way?
So what I try to do is get across where the science is in relation to, if you like, health or applications in medicine and technology. So at the moment, there's one or two drugs that have been developed to target microRNAs. One of them has already gone through a clinical trial. It wasn't for a brain disease. It was for a hepatitis C. It was a liver infection, but it went through to human testing and it shown to work. We've also seen the emergence of some blood type tests. There's a couple of tests, for example, there's a cancer test that's been developed where they measure levels of microRNAs, and that can be used as a way to determine that cancer may be present, a particular type of cancer may be present in somebody. Of course, for us, we're excited that the possibility that you might be able to make a measurement of microRNAs maybe in a blood sample that could tell you if you have a risk of developing epilepsy, or maybe if your epilepsy is of a particular type, or may respond to a particular type of treatment.
That just sounds amazing, just through like getting a bit of a bodily fluid, whether it be a bit of spit, or you mentioned tears before, there are so many potentials here. It sounds like this is, it sounds like your book reflects this, and I can't wait to read it. Reflects that this is an exciting time in this sphere of microRNAs.
Yeah, one of the reasons I wrote the book when I did was because I felt the field had got to a level of sort of maturity. We'd made these incredible discoveries. We'd learned what this system was important for, how it contributed to all of the functions within the body. And we were starting to see applications in the medicine and sort of health technology fields. And in the epilepsy field as well, we'd reached a level of maturity where we understood that these genes really are changing in epilepsy. It's different types of epilepsy. We don't know if it's all epilepsies, but it's certainly many of the epilepsies that we've looked at seem to have some change in the function of these genes. And we're beginning to see the development of potential treatments and diagnostics. But we're not yet getting into the clinic. So will there be a book 2? I hope there might be in the future a point where we can look at where we were now and look back on that time and think that was just before this moved into the clinic, and now look what we've got.
Gosh, if one doesn't get David's book now, then I just don't know. But thank you to David for making learning about genomic history exciting and cool, because that is a brilliant skill in itself. You can find the link to David's book and other info on his work on the website, torierobinson.com, where you can also access the podcast, the video, and the transcription of this episode. And if you haven't already, don't forget to like, comment, and subscribe to the channel. Share this episode with your friends, colleagues, family members, and see you next week.